So again, these are the structures of the protein. So if you see a diagram, this is primary structure of protein. These ones, you see the coiling here. It's like this is secondary structure of protein. So the secondary structures, there are two of them. There is an alpha helix. The alpha helix is the one that coils, and there is plated sheet. Plated sheet is like a sheet of paper. A sheet of paper. So that's another structure. So we have two secondary structures. Alpha helix that's the one that coils, and plated sheet. So both of them are secondary structures. Then we have this tertiary structure, coiling within coiling. Then we have this quaternary structure. So you can be shown these diagrams, be able to identify that this is quaternary structure. You can see for quaternary structure we have two proteins here. We have this protein that is blue, and we have this protein that is faint blue. Tertiary, we have a lot of folding, but it's just one protein. The same thing with helix and the plated sheet. Now let's talk about what is present in the nucleus of cells. So nucleic acids or nucleic acids are polymers present in the nucleus of a cell. So nucleic acids are going to have a phosphate. They must have a phosphate. They must have a nitrogen base. And they might have a pentose sugar. A pentose sugar, it means it is 5-membered ring sugar, like fructose or the like. So there are two types of nucleic acid. We have deoxyribonucleic acid, that is DNA, and then we have ribonucleic acid, that is RNA. So these are the two types of nucleic acids. Two types of nucleic acids. We have DNA and RNA. So what does DNA do? DNA just provides a code, like a blueprint for the formation of amino acids, while the RNA actually forms the amino acids. So DNA is like a planner. This is your plan of the house. Now the builder is RNA. So this one is like a planner. DNA is a planner. This one is the builder. That does the actual job. So these are polymers. But these polymers, DNA are polymers, and the monomers are called nucleotides. So these are the monomers for DNA. So nucleotides are composed of a pentose sugar. It's a sugar but five-membered sugar. So we need to take note of that. Five-membered ring sugar. So a DNA must have pentose sugar, must have phosphate and nitrogen base. Or RNA, the same thing. So DNA or RNA must have these three things. Must have pentose sugar, that is five-membered ring sugar. They must have a phosphate, and they must have nitrogen base. So we'll see what these ones mean. So I'm taking them off here so that I see what we have in the end. I hope you've written them. So this is DNA. So what is this? This part is the phosphate part. This part is the pentose sugar part. Because they can give you a diagram like this to identify it. So all this is pentose, this part. This part is pentose. While this one here is phosphate, where you see the P. This part is the base, the nitrogen base. So if you are asked, can you identify this part of DNA? You need to be able to identify that this part is the pentose sugar, where you see S is the sugar, but mostly really where you see that five-membered ring attached to the PP, that is the pentose sugar. The P is the phosphate part, and these ones on the sides here are the nitrogen bases. And as you can see, even the DNA, what we see here, when we think about DNA always there is pairing. So DNA always has pairing. DNA always has pairing of the chains. You can see here there's one chain here and another chain. DNA comes in pairs. So I say DNA comes in pairs. You can see here chain number 1 here, chain number 2. So DNA comes in chains, in pairs. That's different from RNA. RNA is not paired. Only DNA is paired. So information here. DNA has a sugar, the pentose part. The pentose part of DNA, the pentose part is the sugar part, is called deoxyribose. You will see what I mean when I go to the structures. Deoxy means oxygen has been removed. That's what deoxy means. And the bases that it has, DNA has 4 bases. Cytosine, we just say C. Guanine, we say G. Adenine, we say A. And thymine, we say T. So it has C, G, A, T. What does this mean? Let's go back here. T, thymine. G, guanine. T, thymine. Cytosine, adenine. C, G, A, T. The same thing here. C, G, A, T. So these are the pairs that these are the bases that you can find in DNA. CGAT, cytosine, guanine, adenine, thymine. Normally, the teacher will want you to memorize what pairs are found, what bases are found in DNA. What bases are found in DNA? So the answer is cytosine, guanine, adenine, and thymine. DNA is double-stranded. As I said, it is always in pairs. So we call them complementary base pairing. So the pairing in DNA is called complementary base pairing. So you need to write that down. The pairing in DNA is called complementary base pairing. So what is the difference between the structures of DNA and RNA? The sugar, we start with the sugar. The sugar in DNA is known as deoxyribose. That's why DNA is called deoxyribonucleic acid. The sugar in RNA is known as a ribose. There is no deoxy. What about the bases? The bases in DNA are adenine, guanine, thymine, and cytosine, which is A, G, T, C. The bases in RNA are adenine, guanine. Instead of thymine here, we have uracil. And then we have cytosine. So there is this part. There is one change. So here we have A, G, U, R. And this thing is messing me up and I'm already exhausted. Yeah, so here we have A, G, U, C. So that is, the difference is here we have thymine, here we have uracil as a base. The strands, another difference. DNA is double-stranded with base pairing. RNA is single-stranded. Because DNA is double-stranded, it has an alpha helix. RNA does not have an alpha helix. So write down these differences. Your teacher can ask, what is the difference between DNA and RNA? Just say DNA has deoxyribose, RNA has ribose. DNA has adenine, guanine, thymine. Or just say DNA has thymine in its bases, while RNA has uracil in its bases. That's where the only difference is. DNA is double-stranded, RNA is single-stranded. DNA has alpha helix, RNA does not have alpha helix. So this is what we are talking about. This is DNA. You can see double-stranded here. This is RNA, that is single-stranded. This is a helix, alpha helix. So this one, you can also be given a diagram like this one between RNA and DNA. This diagram, this is a pentose, the sugar. This is the sugar, the pentose, the sugar in DNA. Why is it deoxyribose? You see here, no oxygen here. No oxygen there. You see here, the same place, there is oxygen here. So this pentose here, because there is no oxygen here, is known as deoxyribose. Because we have removed an oxygen here and replaces it with H. Where here we have oxygen on this, so it's known as just a ribose. So this base, this sugar, this pentose here, this pentose, the ribose pentose is for RNA. The deoxyribose pentose is for DNA. Now, when you are given these two structures, look at this one, this place, this position. There is no hydrogen here, it's DNA. Oh, there is no oxygen here, it is DNA. There is oxygen here, it is RNA. That's how you differentiate the pentoses. Every other thing is the same. The only difference is here. So this is the pentose part of DNA. This is the pentose part of RNA. Major functions of RNA is just protein synthesis, builder of proteins. Maker of proteins. Major functions of DNA to form a code, like plan for protein synthesis. So it makes a plan and then goes and tells the plan to RNA that, okay, this is the plan that I want you to do. Use this plan to make protein. That's what DNA does. Playing me in so that I don't have time to finish. So code for protein synthesis or plan for protein synthesis. But it also stores and exchanges genetic material. So all the genes are stored in DNA. Genes stored in DNA. So all the genetic material is stored in the DNA. And then it can transfer those genetic materials from like father to daughter to sons, or from mother to son, from mother to daughter, things like those. That's the function of DNA. Now we have part of DNA that is, we call it adenosine triphosphate. Also part of another nucleic acid. This one, the adenosine triphosphate, ATP, will combine with the ribose of the sugars on the DNA or RNA. So sugars on DNA, actually a sugar on RNA. Let me just, because sugar on RNA is a ribose. So adenosine triphosphate is a molecule that will combine with sugar on RNA to produce energy. So adenosine triphosphate is a molecule that has 3 phosphate groups. A molecule that has 3 phosphate groups. I will show you what these groups are. And yeah, it is used to provide energy to cells. So it provides energy to cells. So the adenosine triphosphate provides energy to cells. In short, we call it ATP. It is actually a high energy molecule. Why is it a high energy molecule? The reason ATP is a high energy molecule is because of the 3 phosphate molecules, 2 of them are easy to break. Therefore, as they break, there is energy that is involved. So what happens is we have the phosphate here, another phosphate, and a third phosphate here. So these can easily break. As they break, energy is released. That's how ATP produces energy for the cell. So here say the terminal phosphate, this one. The terminal phosphate bond is cleaved, it is cut. As it is doing that, it releases energy, and the cell uses that energy being released from the cleavage. So cell uses the released energy. So really, the nucleus is provided energy by the ATP. So ATP provides energy to the cell, including the nucleus. So that's something that you need. What provides energy to the cell? The answer is, yeah, it is the adenosine triphosphate, ATP. So when ATP cleaves, it leaves something that we call adenosine diphosphate, ADP. Adenosine diphosphate. When ATP breaks, it forms ADP. ADP formation produces energy that is used by the cell. So the cell of an animal or the cell of a plant, whatever, gets energy from the breaking of ATP to form ADP. So you can write that down. Since I'm taking it off, just make sure to stop the video and write down the information and then proceed. So energy that is released in the process is used for so many functions. So these are the functions that the energy is used for. Make synthesis of macromolecules, like synthesizing proteins. It is also used for muscle contraction, nerve conduction, and the like. So after the ADP is formed, then the ATP is reformed again. If you add energy in there, you will form the ATP again. So you can add energy from eating food and things like that. So say maybe add energy from eating food. So things like the carbs and the like, they also help in reforming the ATP, because they release energy into the body and the energy will form the ATP. And then the ATP again, when breaking, it will form another energy. So it just forms a cycle. So this is the ATP. You can see it has 3 phosphates here. When it breaks down, it releases energy. It releases energy and forms ADP, 2 phosphates. It forms ADP, which has 2 phosphates, and then this process releases energy. Now, if you put energy back in here, you are going to build up ATP. So it's a cycle. But to build up ATP, you need to add energy to the system. So this is how the molecule of ATP looks like. You can see the 3 phosphates here. This is the first phosphate. This is the second phosphate. So the phosphate comprises the P here and these 4 oxygen molecules or atoms. It's not just the P. P itself is known as a phosphide or a phosphorus. P is phosphorus. But P and O4 is a phosphate. So a phosphate is the whole thing here. Another phosphate is this whole thing. A third phosphate is also this whole thing. That's why it's adenosine triphosphate. Adenosine triphosphate is going to have 3 phosphate groups. Adenosine diphosphate, it has 2 of them. It means that is after breaking the ATP to produce energy, then you have 2 phosphate groups. So that will be the difference. Your teacher may show you these 2 diagrams, the diagram of ATP and diagram of ADP, and say which of these is ATP, which of these is ADP. I think this is where the video ends, and they exhausted me. Thank you, Mama.